Hello! Welcome to another one of my Genetics for Writers and Artists series. This time, thanks to popular demand, we'll be covering dogs. So a few things before we begin. One, this is not a series for dog breeders. This is, as the title suggests, for writers, artists, and so on who are working with fictional dogs. That isn't to say that a prospective breeder couldn't use me as a resource, but... 1. It won't be as helpful to you as breeder-geared resources would be. 2. A lot of specific breeds have wildly different terminology for the same phenotypes. And 3. There's a gazillion genes and phenotypes that are exclusive to specific breeds, but look identical to something else, and I cannot possibly cover all of that. So you'd be better off finding resources for your specific breed than this dogs in general affair. 2. This series expects a baseline familiarity with genetics concepts and terminology. Back when I made my cat series, I didn't expect it to get so popular that people would want another one, so my genetics primer video that you'll need to understand this series is labeled as Cat Genetics, Ep 1. And I can't rename it, it'll mess up the whole series numbering. Whoops! It's not really about cats, it's just a primer of genetics in general that occasionally uses cats as an example, so even though it says cats, and you're here for dogs, you should still watch it. It'll prepare you to understand the rest of the series. And if you're watching this video on the playlist, wondering why the first video on said playlist is Cat Genetics Episode 1, that's why. So watch that, come back, you watched it? Or you already have a solid foundation in genetics? Alright, awesome, let's go. The so-called default color in dogs is black fur with black leathers, which is the usual nose and paw pads, as well as the eye rim in dogs, with brown eyes. Though a black dog can rarely have amber or copper eyes, usually eye color follows the dog's fur color, and black equals brown. A dog that's all black is simply called solid black, but there are various mutations that can cause markings of another color to appear. Brown, tan, fawn, red, yellow, lemon, they're all lumped together into a single category, usually called either tan or red. And for 9 solid hours of my life while researching this video, this was my personal hell! Here's a quick primer on hair and pigment terminology. Pheomelanin is the molecule that causes red and red-based coloration. Eumelanin is the molecule that causes black and black-based coloration. A red hair has pheomelanin, a black hair has eumelanin. Agouti refers to alternating bands of both pheomelanin and eumelanin on the same hair. Agouti hairs can be just pheomelanin, like in orange tabbies, or banded with nothing instead of pheomelanin, like in silver tabbies. But generally speaking, and unless otherwise specified, when we refer to an agouti hair, we're talking about this pheomelanin-eumelanin alteration. The problem is that dog genetics websites use agouti and red interchangeably, as if they're the same thing. More on this later, but these markings are on the Agouti Locus and are called agouti markings, but are also called red markings and referred to as being pheomelanin-based. At first I thought maybe they were red agouti, like orange tabbies, but I couldn't find anything backing that up. As soon as I thought it was just red, and calling it agouti was mistaken, I found this image of an agouti hair from a dog. Then I thought calling it red was mistaken, but no, mutations that only affect eumelanin don't affect these markings, so that doesn't make any sense. I scoured countless websites for hours. I'm not kidding here, cutting out breaks for things like showers and dinner. This was 9 solid hours of my life. And even during that dinner break, I'm not counting, I was pacing around with my soup trying to figure this out. I did not think I was gonna have to break out persona_twitch.png this early into the series, but I was in hell with this. Finally, I got desperate enough to ask a genetics Discord server I'm in if anyone knew what the fuck was going on, and thank God someone replied with the answer. It's just red. It's just red. The locus is called agouti because it's an area where agouti mutations typically occur in other mammals. It's also called ASIP. However, dogs are special, and their mutations in the same area don't cause agouti, except for one. That single agouti hair I saw was from a specific mutation that I'll talk about later. The rest are just red. Imagine, if you will, a world in which all leucistic markings, also known as bicolor or piebald, were called KIT markings because leucistic mutations tend to occur in the KIT region of genes. Then, one weird animal mutates a non-white marking on the KIT region, so we still call the marking KIT even though it doesn't produce white. It would be kinda confusing. Now, I do think this makes sense and is reasonable. It is the Agouti Locus. ASIP is the locus where Agouti happens. Any gene region, no matter what it's most commonly known for, is capable of producing mutations with a wide variety of effects. ASIP is a region responsible for switching between eumelanin and pheomelanin production, so it makes sense that, even though it's much rarer, a mutation here could cause a full switch to pheomelanin rather than the back-and-forth switch of a typical Agouti hair, so calling this locus Agouti is fine. But it would not have been hard for literally any of the dog genetics websites I dug through like a hungry mole for a whole day to contain a simple intro to the air of... This locus is called the Agouti Locus because its gene region usually has Agouti mutations. In dogs, however, only one of these mutations causes true Agouti. The rest simply turn the hairs fully red, with some exceptions to be explained later. And then explaining the exceptions later! I swear, the way these websites talk about Agouti consistently dance around the exceptions being exceptions by just casually mentioning them and making it sound normal, like they expect people to come in with all this background info that no one is giving. I was ranting in frustration to my friend about it, and she compared it to Punt, ancient Egypt's close ally who they did constant trade with. Everyone in ancient Egypt knew where Punt was, and because it was so obvious, no one wrote it down, so now we have no idea where it was. That's how people write about red in dogs. Okay, back to being sane. Get it together, Murphy. There's exceptions, I'll explain later, and I do mean actually explain and not just expect you to already know. But for now, there's eumelanin hairs, and there's pheomelanin hairs. Lots of things I'll talk about later can make the eumelanin in eumelanin hairs look different than black, but the hairs are still just eumelanin. For this reason, we call these hairs eumelanin hairs, and the markings they make up are called black-based. Not always black, but based from a black pigment. There's also pheomelanin hairs. Lots of things can make the pheomelanin look like all kinds of shades, from dark reddish-brown to basically white and everything in between. But no matter the shade, it's still just pheomelanin, so like black-based markings, these markings are red-based, made of pheomelanin hairs. Dog breeders have like a million words for pheomelanin hair markings because every single exact shade they can be each has like 20 names, and people tend to use that instead of an encapsulating word. There's a few encapsulating words. I hear tan used the most. I also hear Agouti sometimes. But here, all shades will just be called red, plain and simple. And I'm going to call the Agouti Locus the A-Locust for the sake of what's left of my sanity. Last note, the genetic server I mentioned is the official server for the Genetic Animals Minecraft mod, which is amazing, and I refuse to play Minecraft without it. The dudes who made it are super cool too. Check it out, link in description, download it, show off all your genetically accurate Minecraft dogs in the Discord, you won't regret it. Okay, we're finally ready to talk about some actual genes. The K locus has 3 alleles. The KB allele causes dominant black, in which the dog is just totally solid black with no red markings. Obviously being called dominant, it is at the top of the K locus's dominance order. Fun fact, black North American wolves get their coloration from ancient hybridization with domestic dogs. It's the same KB allele, but instead of dogs getting it from wolf ancestors, wolves got it from dog ancestors. Well, we don't know for sure if the European wolf ancestor that all surviving dog breeds descended from had it or not, but existing European wolves don't anymore, and North American wolves didn't come in black at all until domestic dogs came along and gave it to them. According to a 2009 study, black-inducing KB is actually receiving some positive selection in forest environments, aka evolution is preferring black wolves over non-black in Northern American forest regions. Anyway, after that is Brindle, represented by Caber. So when a body makes new gametes, eggs or sperm, all the homologous, aka paired chromosomes, do this thing called recombination, where they swap loci with each other. The A locus gets swapped with the other A locus. The K locus gets swapped with the other K locus. This is normal, this happens. So here's a chromosome pair. Let's pretend that this is the K locus. It's not even slightly to scale, it's just here to make a point. It doesn't need to be accurate. Left chromosome has a KB allele, right chromosome has a KY allele. One day, in some dog thousands of years ago, they went to do their normal recombination, but something went wrong. Hey, you got your KB and my KY. You got your KY and my KB. It looks great. 2 great dogs that look great together. Reese's, Brindle puppy cup. They ended up getting stuck to each other on the same chromosome, is what I'm saying. Anyway, the gene is unstable. Actually explaining what a transposon is not going to happen here, I'm sorry. Link in the description to a video that can show you a little bit about what they do because it's super interesting, but I barely understand them myself, so study those in your own time. The seriously simplified and thus inaccurate, even more so than usual summary is that Caber's two halves really don't like being stuck together. In a single dog who has the Caber allele, every single cell in that dog's body has the Caber allele because every cell contains the entire genome, right? So individually, inside of each single cell, the Caber allele can either stay a Caber allele, or it can lose either its KB half or its KY half. If it loses its KB half, then it's just a KY allele and it makes the dog red. If it loses its KY half, then it's just a KB allele and it makes the dog black. So there's some cells producing red, some cells producing black, and the dog ends up a mottled mix of both. It's like Garnet from Steven Universe, right? She's Ruby and Sapphire stacked on top of each other in a trench coat, creating a whole new type of guy. But sometimes they argue, and they split up, and one of them leaves. If just Ruby's here, the cell is red, and if just Sapphire's here, the cell is black. Caber, being what my friend described as silly style, can also double KB instead of kicking out KY. One of my sources calls this new extra tall allele Caber Plus and suggests that it might be part of the reason some brindle dogs are mostly black. No evidence for this yet, though, just an idea they put out there. Following our metaphor, this is like if Ruby and Sapphire let a second Sapphire into a polyamory, I guess? The Caber can also not change at all and just stay Caber. I can't find anything saying this directly, but from what I can glean from other information, I think when Caber stays Caber, it expresses black? Don't quote me on that. Anyway, if these splits or duplications happen in places where gametes are created, the testes or the ovaries, then there's a chance that those gametes could contain KB, KY, or Caber Plus instead of Caber. So even a homozygous Caber dog can end up passing KB or KY to their offspring instead. Or they just pass normal Caber, that also happens. Fun fact, wild corn also has a transposon responsible for some of its coloration, and that's why it gets all mottly like this. This is the corn equivalent of Brindle. These types of patterns that result from differences in expression between individual cells are called mosaics, and they don't always result from transposons. Tortoiseshell cats are mosaics because of second X deactivation instead, for instance, but that's a different video. Finally, fully recessive to both KB and Caber is KY, which I mentioned in passing during that transposon explanation. It probably sounded like KY just turns a dog red, but it's more complicated than that. It's the light switch locus that allows us to see the A locus, which is where all those red markings are. So what KY does to the dog depends on what the dog has on their A locus, aka the A locus is a modifier of the KY allele. The A locus also modifies Caber, by the way. If a dog is KY, then we'll see whatever red markings the A locus codes for. But if the dog is Brindle, then we will instead see whatever red marking the A locus codes for, but all the red areas will be brindled instead of solid. So while I talk about the A locus, I'll show you side by side what the markings look like with KY and what they look like with Caber. With the exception of a gene exclusive to border collies that we don't know enough to talk about yet, the A locus has 6 alleles, and each allele codes for a different marking. As for dominance, it's generally accepted that all of the A locus alleles, with one exception, are incompletely dominant with each other. Any heterozygote displays a marking somewhere between the markings that both alleles cause. Though for some reason, some breeds get a heterozygote phenotype that's more evenly mixed, and some breeds get a heterozygote phenotype that leans more towards the quote-unquote more dominant allele. No idea why. So there's still an accepted dominance order, and I'll explain the alleles in that dominance order. Here's some important terminology. In cats, tipped refers to when a single hair has a little bit of pigment at the top, and the rest of the hair is a single long band of something else. In dogs, however, that is not what tipped means. Tipped in dogs means that the dog's body mostly has entirely pheomelanin hairs, but a couple of stray, entirely eumelanin hairs hang out up near the spine, top of the head, or the ears. This type of hair, with the differently colored tip, is instead called Sable. The markings produced by the alleles I'm about to talk about are also called Sable because they contain this type of hair, and sometimes they contain tipping. Sable is one of those weird exceptions, by the way. It's technically an agouti hair, but it's a weird one. There's 3 types of phenotype called sable, as opposed to sable hairs. Clear sable, tipped sable, and shaded sable. The AY allele causes clear sable. Clear sable dogs are basically totally red, they generally don't have any tipping, or if they do, there's very, very, very little, and they have a few sable hairs, but there's so few that we can't really see them. It's important to keep in mind that no matter how little black base is actually in their coat, the dog's leathers and whiskers will still be black-based. So even a clear sable dog's nose will be black, or one of the things that modifiers can turn black into. In fact, you can tell which black-based color a red dog is hiding by looking at their nose. More on those in future videos. Until recently, we thought that AY also caused tipped and shaded sable as well, but it turns out that at least tipped is caused by a different allele. The website I got this one from is a gene testing lab, so it uses more complicated science-y names for the alleles that are very different from the A-something nomenclature. They call it ASIPsy. I'm going to shorten that to ASY here for the purposes of fitting into the rest of our established nomenclature. Caused by ASY, tipped sables, as the name suggests, have more noticeable tipping. This gives a kind of grizzled black-based look along the spine, and sometimes parts of the ears go black too. Lastly, there's shaded sables. Shaded sables have the highest amount of stray hairs, often to the point where you can't really call it tipping anymore. There's a lot of variation, but keep in mind while drawing that they always form a kind of widow's peak marking at the forehead. We don't know exactly whether shaded sables are ASY being affected by polygenes that increase tipping, or if it's AY or ASY being heterozygous with something else, and the incomplete dominance is causing more eumelanin hairs than we might otherwise expect from AY or ASY on their own. No reason the two explanations can't both be right. By the way, these black masks are totally unrelated to the K and A loci, so ignore them for now, and I'll get to them in a future video. After all those sables is AW, which causes, unless we count a handful of stray sable hairs, the only actual agouti on this locus. That single agouti hair that made me lose my shit is from this marking. This is the one allele on the so-called Agouti Locus that is actually defined by agouti. I don't know whether to sing hallelujah or cry. To avoid confusion, because the locus is technically also called agouti, I'll be referring to the marking caused by AW by its alternate name, wolf gray. This is the wild-type gene. This is the one that wolves have, that dogs retained after domestication. Without a magnifying glass to look at individual hairs, wolf gray looks very similar to shaded sable. However, the layout of the marking is different. With wolf gray, we don't really see the widow's peak that shaded sable has. Instead, we see a bar of black going up the nose to the top of the head. We also see spectacles of red around the eyes. Wolf gray dogs may be born dark and then lighten as they age. Fun fact that makes me want to pull my hair out. Wolf gray has a third name. For some ungodly reason, German Shepherd breeders decided to call wolf gray Sable. Dog breeders just do not communicate with each other, apparently. ABS, or ABS if you want to pronounce it, which I do because it's funny, is another one of the recently discovered genes alongside ASY. It's actually called ASIPbs, but again, I'm shortening it to fit our nomenclature. ABS causes the saddle marking, which is the clear patch of black over the back, shoulders, and often up the neck of the dog. This is the classic beagle look. After ABS is AT, which causes tan points. This is an iconic dog marking and also one of my personal favorites. It makes very distinct, sharp red markings above the eyes, around the muzzle, and the top of the neck, triangle-ish spots on the chest, red paws, and red on a good chunk of the legs too. If any black remains on the toes, that is called penciling. A lot of dogs who look like they have white markings are actually just pointed dogs with a ton of modifiers that make their red look super, super light. Actually, that's true of a lot of A locus markings, but it's especially easy to mistake pointed for piebald. And actually, I lied to you, there's 3 variations of AT. ASIPBB1, ASIPBB2, and ASIPBB3 all do the same thing and inherit the same way, so we group them together and pretend they're a single allele called AT for simplicity. Also, for some reason, scientists call the pointed phenotype black back, which is not only a tongue twister, but also describes almost all of the phenotypes that the A locus causes, so that's dumb. I'm sorry, fellow scientists, but name your markings better. Finally, we have recessive black, which is little a. Now, unlike all the other alleles on A, which are usually incompletely dominant with each other, recessive black is what it says on the tin. It's fully recessive. It's also pretty rare. It does the same thing that KB does, wherein the dog becomes fully black, taking us full circle. That means a KY dog can be solid black, even though they have no KB alleles. Crazy, right? When I say black here, I'm of course referring to black base, hairs with just eumelanin in them. That means any genes I cover in later videos that modify black/eumelanin will also affect recessive black. It's the same phenotype, just caused by different genes. That's the end of the A locus. One more gene before we get to the practice squares, though. Both tan points and wolf gray can be modified by a separate gene on a separate locus called rally, which causes creeping tan. If a pointed or wolf gray dog has creeping tan as well, then the dog is born with a normal pointed or wolf gray pattern, but as they age, the red pattern expands upward until the black retreats. Once the red is past the head, it starts being, confusingly, also called saddle. So saddle can be caused by the abs allele, in which case the dog is just born already looking like that, or saddle can be the later stages of creeping tan, wherein they started pointed or wolf gray and turn into saddles over time. I don't know how this affects dogs who are heterozygous for pointed or wolf gray and something that creeping tan doesn't affect. My best guess, and this is purely a guess, is that it possibly affects them but not as much, but I have no idea. Apparently rally is called the AS locus, which is confusing and I hate it. It's dominant and uses AS. The allele that doesn't cause creeping tan is called N. Now for some practice squares. We'll do one for K and then one for A, the same pair. If you're looking to challenge yourself with a multi-loci Punnett square, here are their genotypes. This is a black dog and a brindle pointed dog. For simplicity, the brindle here is just passing on normal caber. For the rest of you, here's the first square. In fact, now that the square is on the screen, I don't even need to tell you to assume that caber is just passing on normal caber. The way a Punnett square is written already assumes it for you. A Punnett square is just a visual way of writing out gametes fusing post-recombination and meiosis. So if this brindle is passing on KB or KY, the square would say KB or KY. It doesn't, it says caber, so the alleles in the gametes are in fact normal caber. Okay, here we go. Pause now. Nice. 50% chance of a solid black dog, and 50% chance of a brindle dog. Now let's see what shape that brindle takes by examining its modifier. Here's the next square. Little tricky interpreting the phenotypes here, but if you got the letters right, you got it right. Given the incomplete dominance between ASY and AT, this dog will most likely be a shaded Sable, though again, the incomplete dominance seems to vary from breed to breed. Either way, we'll get something somewhere between tipped Sable and tan point, leaning towards tipped Sable. It's just a matter of how much it leans towards Sable. And that was my personal hell for the last few months. You can see why this series has taken me as long as it has to get started on, I hope. Next video is going to cover one of Black's modifiers, and will be much simpler than whatever the fuck was going on in this video. I'll see you there.